name is Ugochukwu Maluze from University of Sydney, Australia. The title of my presentation is Regulation of Muscle Proteins and How Mutation in Muscle Proteins Causes Heart Disease. Regulation of muscle contraction and how mutation in the muscle protein causes heart disease. The laboratory techniques include molecular biology, protein expression, purification, and characterization using biochemical and biophysical techniques. Next slide. Introduction. Cardiomyopathy has evolved deep attention with increasing understanding of its prevalence, its function as the main source of unexpected cardiac death in juvenile individuals, and its isolation as a Mendelian autosomal dominant genes. Cardiomyopathy can be defined as a clinically diverse group of heart muscle ailments which has a distinguishing feature of unusual myocardial structures. It is characterized by irregular septal hypertrophy, efficient aortic stenosis, hypertrophic osteoporosis, cardiomyopathy, and idiopathic hypertrophic subaortic stenosis. Genetic mutations involved in cardiomyopathy. The genetic mutations in the muscle protein can cause different types of abnormalities in genetic compositions of muscle protein. Such mutations include point mutation, which has been noted to cause hypertrophic cardiomyopathy and often caused by chemicals or malfunctioning of DNA replication and exchange of a single nucleotide for another. Splice site mutations or reading frame mutation is a type of gene mutation that occurs due to insertion or deletion of the genetic element in the coding area of the genes, and this can change the splicing of the messenger RNA, which is known as split site mutation, or lead to a change in the reading frame, known as frame shift mutation. These two mutations can extensively alter the genetic component, alter the protein structure, and contribute to cardiomyopathy. The two mutated proteins that is known to cause or initiate cardiomyopathy are D175N, which is a specific mutation in the alpha tropomyosin protein with amino acid at position 175 that changes from aspartic acid to asparaginase. The second mutated protein that is recognized to cause genetic mutation that affect the heart is known as E180G, which is a specific mutation in the alpha tropomyosin protein that is crucial in muscle contraction. Types of cardiomyopathy. There are 4 known types of cardiomyopathy. Familial hypertrophic cardiomyopathy. Based on phenotypic and clinical examination of the affected individuals, familial hypertrophic cardiomyopathy occurs as a result of autosomal dominant heart illness with prominent features of interventricular hypertrophy, mitral valve disorders, monocyte hypertrophy, interstitial fibrosis, and atrial fibrillations. The illness starts as a benign condition and progresses to a severe devastating state that gives rise to sudden death in young athletes. The structural proteins that causes familial hypertrophy are beta and alpha myosin proteins, troponin T proteins, myosin binding protein C, light chain and troponin proteins. Dilated cardiomyopathy. This occurs as a result of genes alteration of protein tropomyosin, and it occurs in families with autosomal dominant inheritance prototypes with autosomal recessive inheritance. It is characterized with enlarged chamber size and abnormal systolic reduction of the two ventricles. The abnormal proteins involved in dilated cardiomyopathy are sarcolemma and sarcome proteins. Atherogenic right ventricular cardiomyopathy. This occurs as a result of genes mutation on the protein tropomyosin, and it occurs in families with autosomal dominant inheritance. The abnormal proteins that causes such mutations are desmosomal proteins, plakophilin 2, and plakoglobulin proteins. The fourth types of cardiomyopathy is known as restrictive cardiomyopathy. This condition is linked with abnormal ventricular diastolic features with a raised end diastolic pressure that give rise to lower ventricular magnitude. The diagnosis of restrictive cardiomyopathy is very poor in affected children, and heart transplantation is frequently performed. The abnormal protein that causes the disease is cytoskeleton protein desmin. Biochemistry of tropomyosin, its characteristics and definitive role in muscle contraction. Tropomyosin is a protein that plays a structural and active function with cardiac actin in sarcomeric muscle protein. Biochemically, it comprises of 14 exons and 4 isomes. These straighter muscle isoforms consist of alpha helical proteins. The polypeptide chains of tropomyosin contains 280 amino acid residues, and each of the dimer is attached to 7 active monomers and 1 troponin complex. Aims and objectives. The aims and objectives of this research was to produce a wide type of normal proteins known as tropomyosin and mutated genetic proteins, which I have mentioned. There are 2 mutated proteins, E180G and D175N, which are clinically involved in familial hypercardiomyopathy. Having in mind that the main effect of mutations on E180G and D175 proteins are mainly related to the thermal stability of the proteins, this research was conducted as well to investigate the differences between the thermal stability of wild-type and mutated proteins using a dye-based fluorescent method of analysis. The materials involved was 50 mL Fisher-Brand tubes, a Pendorf centrifuge tube, thermoscientific gene DNA ladder, column chromatography fraction collector, liquid chromatography controller, and etc. Methods. The method involved in the analysis was preparation and storage of competent L, XL1, BL2, and E. coli cells applicable to transformation and transfection. This involves transferring XL1 and BL2 cells, which plays certain roles in the analysis, into 2 different conical flasks containing the culture media, AB media, and incubated the cells for 4 to 5 hours at 37 degrees to produce competent cells. The two cells, XL1 and BL2 cells, undergo series of centrifugation. The supernatant was discarded and the pellets were kept on ice for 5 minutes. The cells pellet on XL1 and BL2 were resuspended, pre-cooled ice and mixed thoroughly. About 100 microliter each of XL1 and BL2 cells were resuspended in epinephrine sterile tubes, followed by addition of 2 microliter of DNA to each of the cells and kept on ice for 15 minutes incubation, subject to heat shock at 42 degrees centigrade. About 900 microliter of LB medium was added to each of the tube containing the XL1 and BL2 cells, and heat shock for 30 minutes for transfection reaction was conducted. About 10 microliter of ampicillin antibiotics was added into the solid agar media and mixed properly. The solid media of 6-plate media, which consists of 6-plate media, was allowed to cool, solidify, and gel. About 100 microliter of XL1 and BL2 was added to the first and second plate, spread uniformly to make distinct colonies, and XL1 and BL2 low cells concentrations as well. To obtain high concentration of cells of the XL1 and BL2 cells, these cells were spun for 1 minute and resuspended. About 100 microliter of XL1 and BL2 spun cells were added to the third and fourth media plate, respectively, and spread uniformly, and labeled third and fourth high cell concentration. Furthermore, about 100 microliter of XL1 and BL2 cells deficient of DNA was used as a negative control and added to the fifth and sixth media plates, respectively, and labeled clearly XL1 and BL2 negative control cells. The sixth media plate, which is labeled as XL1 and BL2 low cells, XL2 and BL2 high cells, and XL1 and BL2 negative control, were incubated overnight at 37 degrees centigrade to reach the colony. To grow the BL2 culture cells, a single colony was incubated to each of the 2 sterile epinephrine tubes containing 1 microliter of each of LB, ampicillin, and the 2 tubes were labeled. To analyze the non-induced and induced proteins on the gel electrophoresis, about 5 microliter of the DNA ladder was loaded on the first well of the ladder. 15 microliter of non-induced BL2 cells were added to the second well, and 15 microliter of induced BL2 cells were added to the third well of the ladder. 15 microliter of non-induced XL1 cells were added on the fourth well. 15 microliter of induced XL1 cells were added to the fifth well. Samples were run at 240 voltage power source and 40 milliamps for 50 minutes. The protein band formation was analyzed carefully. Tropomyosin preparation. The tropomyosin protein was prepared through the centrifugation and sonifications and protein denaturations. The supernatant was transferred into 2 epinephrine tubes and labeled post-boiled proteins. DNA purifications. The pellet the pilated bacterial cells in the second tube was resuspended in 250 buffer P at 40 degrees centigrade and transferred to a microcentrifugal tube. Column chromatography analysis of tropomyosin normal proteins. The protein samples was purified using column chromatography method of purification, and the purified protein were collected on 24 fraction tubes, which was labeled from tube 1 to 24 to tube 24. Protein gel electrophoresis was carried out by selecting the even numbers on the labeled tubes containing the purified proteins. The even numbers that was labeled was 10, tube 10, tube 12, 14, 16, 18, 20, 22, and 24. Each of these tube of proteins then 20 microliter of SD buffer and 20 microliter of distilled water was added into each of the proteins. 5 microliter of DNA ladder was added on first well, and sample from second tube was added to the second well. Preparation of mutated protein D175 and E180G, known as site-directed mutagenesis. To prepare for this mutated protein, 1 microliter of DNA polymerase was added to sample reaction, and including 1 microliter of restrictive enzymes, which was mixed thoroughly and undergo for centrifugation for 1 minute, incubated at 37 degrees centigrade for 1 hour to digest the parental supercoiled DNA. The 4 tubes were labeled non-induced and induced D175 proteins and non-induced and induced 180G proteins. Gel electrophoresis was carried out by loading 5 microliter of the ladder on the first well and 10 microliter of each of the protein sample to the subsequent wells of the ladder to observe the protein band separation of non-induced and induced proteins. Column chromatography analysis of the mutated proteins. The mutated protein samples of D175N that undergoes protein gel electrophoresis was analyzed using column chromatography method. Measurement of the thermal stability of wild-type proteins known as lysosomes and mutated proteins known as D175N and E180G using dye-based fluorescent method. The principle of the assay. The protein is mixed with the dye and heated. As it unfolds or melts, hydrophobic parts of the protein are exposed and bind to the dye, resulting in a significant increase in the fluorescent emission detected by the PCR system. To demonstrate the effect of various buffer concentration of tropomyosin, about 16 PCR tubes were added to each of the tube 1 to 12. 45 microliter of protein samples were added to tube 1 to 12. 40.3 microliter of buffer were added to tube 1 to 3 of protein samples. 40.3 microliter of buffer 2 were added to tube 4 to 6 of protein samples. 40.3 microliter of buffer 3 were added to tube 7 to 9 protein samples. All these, they have different buffer concentrations, which demonstrate the thermal stability of the proteins being analyzed. 2.5 microliter of lysosomes plus 42.5 microliter of buffer 1 were added on tube 13 to 14 protein samples, which is used as a positive control. Then 45 microliter of buffer 1 were added to tube 15 to 16, which contains the protein samples that was used as a negative control. To demonstrate the effect of one buffer with various concentration on mutated proteins and wild-type normal lysosome proteins, 3 PCR tubes were arranged and carefully labeled tube 1, tube 2, and tube 3. 6.03 microliter of E180 G proteins was added on 39 microliter of buffer 1 and 5 microliter of dye to tube 1. They were mixed thoroughly and was loaded on PCR machine for thermal stability. 6.24 microliter of mutated protein D175 were added to 38.7 microliter buffer 1 and 5 microliter of dye into tube 2. They were as well mixed thoroughly and loaded into the PCR machine for thermal stability measurement. 7.2 microliter of normal proteins lysosomes were added with 37.7 microliter of buffer 1 and to 5 microliter of dye, which were added to tube 3 and mixed thoroughly and was put inside the PCR machine for thermal stability measurement. The result analysis. On the next slide is the result. The above diagram on the result shows the XL1 cell that has been cultured with LB agar media. It shows the XL1 cells cultured on LB agar media. There was scanty growth of E. coli, which was observed on the two plates due to ampicillin antimicrobial sensitivity. The E. coli was able to grow on the culture plate because of the XL1 cells containing a vector DNA gene. This gene facilitates for colony growth of the bacteria, which has different morphologic appearance and diagnostic features. The next slide is the negative control of the XL cells cultured on the same LB agar media. It indicates a negative control cell cultured on the media. There was no prominent growth of E. coli because the incubated cell was deficient of the vector DNA. This was used as a negative control method to observe the effect of absence of vector gene on the growth of E. coli. The next slide is low and high concentration of BL2 cells cultured on LB agar media. The next slide is the negative control of BL1 cells cultured on LB agar media, showing no bacterial growth due to absence of genetic vector. The next slide describes the two slides. The above diagram shows the BL2 cells cultured on LB agar media, which was this slide. Then this is the description of the cultured media plate. Moderate growth of E. coli were observed on the two plates due to ampicillin antimicrobial sensitivity. The E. coli was able to grow in the culture media because the BL2 cells contain a vector gene which contains the antibiotics that has the promoter gene component. The promoter gene will bind to the IPTG and stimulate the messenger RNA to synthesize protein component. The next slide shows the purified DNA gel electrophoresis showing double band formation of DNA. The confirmed DNA will transform the BL2 cell to start synthesizing the normal proteins. The next slide is the observance of the tropomyosin normal protein using spectrophotometer measurement. The diagram refers to the observance of tropomyosin normal proteins. The molecular weight of the protein, which can be found on this table on the slide. The molecular weight of the protein was found to be 32,797 Daltons. At 280 nanometer wavelength, the absorbance of the protein was 0.125, and the protein concentration was found to be 11.64 milligram per ml, as found on the table of the slide. The next slide shows the protein gel electrophoresis of non-induced and induced BL2 cells and non-induced and induced SL2 cells. The first well, if you can look at the slide, the first well is a ladder with a molecular weight of 1 kilo base pairs. The second well indicates a double band formation of non-induced proteins, such as BL2 samples, while the third band shows a thicker protein band of BL2 cells, indicating a high synthesis of proteins. The fourth well also shows a non-induced protein band separation of L1 cell samples, and the fifth band shows a thicker protein band, indicating a high synthesis of protein XL1 cells. On each sample well, the protein band occurs between 760 to 780 base pairs. The next slide shows the normal protein pellet before resuspension with potassium phosphate, and the normal protein pellet after resuspension with protein phosphate. The figure indicates the diagram indicates the resuspension of protein pellet with potassium phosphate solution. The pellet became cloudier on the addition of potassium phosphate as a result of protein and DNA precipitation. If you can watch on the slide, you can see that the color of normal protein before resuspension with potassium phosphate is not the same color of protein after resuspension with potassium phosphate. So, which indicates that the addition of phosphate as additional phosphate as a result of protein and DNA precipitation. The next slide shows the protein fraction gel electrophoresis after chromatography purification analysis. The figure shows the gel electrophoresis of purified protein fractions. The first well from the left-hand side is the DNA ladder with a molecular weight of 1 kilobyte Dalton. The highest protein purification was collected on tube 23 and 24, which has a paired protein band formation. The next slide shows the protein gel electrophoresis of non-induced protein, induced protein, post-boil protein, and fractions of 10th, 12, 14, 16, 18, 20, 22, and 24 fractions of normal protein as labeled on the test tubes. The next slide explains the gel electrophoresis. Referring to the diagram above, the first well from the right-hand side, which is this, is a ladder which has a molecular weight of 1 kilodalt and 1000 base pair. The second well, which is this, is a non-induced protein band which has a slight protein band concentration. The third and fourth wells are the induced and post-boil proteins which were induced by the addition of IPTG. Their protein bands were thicker when compared with non-induced protein bands, which indicate an increased synthesis of protein as shown on the gel electrophoresis ladder. The fifth and sixth well shows the fraction collection of purified protein on tube 10 and 12, which precipitate as a double protein band, as shown on the gel electrophoresis well. The seventh and eighth well shows an increase in protein purification fraction on tube 14 and 16, which precipitate with a thicker protein band. The ninth and 10th well shows the fraction collection of purified protein on tube 18 and 20 with a reduced protein band formation. The 11th and 12th well shows the fraction collection of purified protein on tube 22 and 24 with a high precipitation and formation of protein band. The precipitation and formation of protein band is directly proportional to the purity of the protein collected on the fractionating tubes. The next slide shows the purified DNA gel electrophoresis, showing double band formation of DNA. The next slide shows the above diagram refers to the observance of mutated D175 proteins using electrophotometer measurement. The molecular weight of the protein was found to be 32797 Dalton. At 280 nanometer wavelength, the observance of the protein was 0.032 and the protein concentration was found to be 5.25 milligram per ml as shown on the diagram. The above diagram on the next slide refers to the observance of D179 protein using the spec measurement. The molecular weight of the protein was found to be 32797 at 280 nanometer wavelength. The observance of the protein was found to be 0.032 and the protein concentration was found to be 5.25 milligram per ml. The diagram refers to the observance of E180 mutated protein using the spec measurement. The molecular weight of the protein was found to be 32797 Dalton. At 280 nanometer, the observance of protein was 0.031 and the protein concentration was found to be 5.53 milligram per ml. The next slide refers to the gel electrophoresis of D175 and E180 mutated proteins. The first well from the left-hand side of the ladder is the ladder which has a molecular weight of 1 kilodal and 1000 base pair, as can be seen on the ladder. The second and third wells are the D175 end, which serves as non-induced protein band, and D175 end, which serves as induced protein band, respectively. The fourth and fifth wells were the post-Boyle samples of the mutated D179 and E180 G proteins. Their protein bands correspond to the base pair between 960 to 980 kilobytes of the ladder. The sixth and seventh wells are the first and the seventh D175 N pellet with a very prominent protein band concentration. The eighth and ninth wells are shown on the ladder. The eighth and ninth wells are the first and second E180 pellet, and the tenth well is the E180G, which shows the induced protein band formation. The next slide shows the buffer stability of tropomycin and lysosomes at different buffer concentration. Increasing the phosphate buffer concentration causes a 3 to 10-fold increase in the thermal stability of proteins. The diagram shows the effect of the same buffer with different ionic concentration on tropomycin, mutated proteins, and lysosomes. The buffer has a varying concentration at the same effect. An increase in phosphate buffer concentration increases the conformation and unfolding of proteins. By merely looking at the graph, the labeled well known as UN care 1, 2, and 3 indicate the normal type tropomyosin. UNK 4, 5, and 6 symbols indicate the D175 mutated proteins, and the UNK 7, 8, 9 symbols indicate lysosome proteins. The NTC1 symbol indicates the negative control sample, which contains the dye without the protein component. In conclusion, recombinant human tropomyosin were produced, which serves as a model protein to study the interaction that govern the stability of the thin filament. Two mutations, E180G and D179 mutated proteins, described as causing cardiomyopathy, were introduced in the DNA encoding the human skeletal muscle tropomyosin. Thermal denaturation studies of control and mutant tropomyosin shows a cooperative interaction between folding of the N domain and C region of tropomyosin. The hypertrophic cardiomutations on E180G and D175 prominently destabilizes the tropomyosin N domain, which indirectly affected cooperative interactions between the N and C terminus regions of tropomyosin. The experimental result on this current study that the hypercardiomyopathy mutations locally destabilizes tropomyosin, leading to an increased thin filament caching sensitivity and the degree of disruption of the tropomyosin interaction by the cardiomyopathy mutations may be directly correlated with the severity of the disease. For future prospects, future biochemical analysis of several other hypercardiomyopathy mutations will be needed to establish a definitive correlation between the enzymatic impairment between different mutants and their clinical phenotypes of the heart disease. These are the references, and the link to this publication can be found on this link on the slide, which is the last slide. You can find the links to this. Thank you. If you have any question, you can ask.